Right, so for no other reason than we do have a bus to catch, I shall keep the introduction relatively brief. That's no reflection on our esteemed guest and Bartlett lecturer, Marlene. If you didn't know, sorry, that might be a spoiler, sorry. Just a bit of background on the award itself. So the Sir Frederick Bartlett Lectureship is endowed by a trust from the said man. It was first delivered in 1966, which holds rather pleasant memories for those of us who are football fans, but probably not for many other people. I was 4 at the time, I can hardly remember it. So the Bartlett Lecture itself is awarded to someone who has made a distinguished contribution to psychology or cognate discipline. This year's Bartlett Lecture, as I've already given away, is Marlene. And you will have heard across a brilliant series of talks over this afternoon what her contribution has been, both in cognitive psychology, in experimental psychology, in computational modelling, in cognitive neuroscience. The list goes on. It's quite incredible. A little bit of background. Marlene did her PhD. Is it okay if I say when you did your PhD? Yeah, okay. I should know. I wonder these days sometimes. So Marlene did a PhD in 1991. She's had positions at Toronto, Carnegie Mellon, and most recently at Pittsburgh. I went through a CV earlier, sorry, I hope you don't mind. I was going to read out a prize or two that she had already won, aside from this prestigious award, and it would take me rather too long to go through them all, and it would significantly impinge on both our ability to hear the guest speaker, but also Bartlett lecturer, but also to catch the bus. So I think it's rather, both of those things are not equally important, but they are both important. So I hope you will now join me in welcoming Marlene to the stage to give her Bartlett lecture, the 52nd Bartlett lecture. And that's not length, it's going to be about 50 odd minutes. So, welcome, Marlene.
Thank you very much for that kind introduction. I am grateful to Carolyn for putting together the symposium. I'm grateful to the EPS and to all of those who assisted with today's logistics. I'm also deeply honoured to present the Bartlett Lecture, and I'm humbled to join the distinguished group of investigators who have presented Bartlett Lectures before me. Among the most rewarding aspects of doing research is the opportunity to work with others, and as you see for me, the distinction between students, collaborators, friends, and colleagues all kind of merge together and blur. And so I want to thank my colleague friends who just presented in the symposium. I also want to draw specific attention and express special gratitude to Carolyn Patterson and to Dr. Patterson and Dr. Sally Bing. Carolyn sent encouraging letters beautifully penned on blue aerogram paper when I was a young speech therapist in South Africa. And she opened my eyes to the possibility of studying patients from an experimental viewpoint. When I subsequently studied at Birkbeck College, London, under the auspices of a British Council fellowship, Sally was my role model of a speech therapist becoming a PhD investigator. And I'm really pleased that she could join us today. I'm also really glad to see so many friends I've made along the way in the audience. I'm not going to go through and mention all of you, because I know very many here, but I really do want to specifically point out that Steve Tipper, my collaborator in the work that Jay presented today, was able to be at this talk, and I'm so pleased to see him. I also want to recognize the contribution of 29 postdocs and 19 graduate students with whom I have worked, many of whom have contributed to the work that I will be presenting today. Confronted with a complex visual scene such as this one, with just a little time to inspect, you can undoubtedly see that there is a Pope-like character being drawn on a white horse, that there is a woman here holding out a flower, and then there is this man here with a rather impressive sword and shield. Because we can perceive this untold richness with such precision and seemingly effortlessly, it was long thought that we carried a camera around in our heads. Well, we don't. But what we do carry around is a visual system that occupies roughly 60% of the brain, comprised of 4 to 6 billion neurons that are organized into roughly a dozen distinct areas that cover occipital, temporal, and parietal cortex. How does the brain support this remarkable ability to recognize? So in today's talk, I'm going to address a number of questions, 3 of which are here. How is visual recognition organized within and between the cerebral hemispheres? How does this ability emerge over development? And how malleable is this ability, or is this form of organization following breakdown? To the naked eye, the 2 hemispheres of the brain are remarkably similar, aside from some small anatomical differences. But decades of research attest to the substantial functional differences between the hemispheres. Now, the popular press is exceptionally excited about these hemispheric differences and would have us believe that the distinction is between a right hemisphere that is colorful and joyous with guitar playing and kite flying capabilities. In contrast, the left hemisphere seems to consist of faceless people working in cubicles, toiling in the pursuit of calculation and inquiry. Sounds a little familiar. But we don't have to believe the popular press, of course. We can, in fact, depict the topographic organization of the hemispheres by having participants view images of words, faces, common objects and scenes or places while lying in the bore of a magnet. And then afterwards we can calculate the amplitude of the selective response to these different categories in different regions of the brain. So you've already seen the image that I'm going to show you, but the standard result is that there is uniform topographic organization with different areas responding to different stimuli. So I'll just warn you here that the right hemisphere is going to be projected on the left of the image and the left hemisphere on the right of the image following radiological conventions. I've also tried to always label it just so that we don't go astray in the process. So you can see here there are these domain selective areas, the fusiform face area for faces, the visual word form area for words, and then bilateral activation for places in the parahippocampal place area, and a huge swath of cortex shown here in blue in the lateral occipital regions that is selective for common objects. And this large swath is probably the result of the fact that the class of common objects contains so many objects with highly differentiable geometries. I'm going to take as the domain of my investigation today this binary segregation between the right hemisphere fusiform face area and the left hemisphere visual word form area. So there are good a priori reasons to claim that this segregation is an optimal organization for the visual recognition system. Firstly, the disparity in the image statistics between faces and words is so vast, this might compel an independent representation for these different stimulus classes. It's also the case that there is a substantial difference in the evolutionary recency of these stimulus types. So whereas face recognition has been around for humans for an extremely long time, word recognition has really only been dominant in the human culture for roughly the last 150 to 200 years. And last, whereas face representations appear to be acquired incidentally, starting from birth, word recognition is typically acquired through explicit instruction when children start attending school. All of these reasons, theoretical claims, support and favor the independence of word and face systems. But there is one further argument, and that is that because the class of words and the class of faces each have so many homogeneous exemplars that require fine-grained visual discrimination, the segregation of these two systems would allow for fine-tuning to optimize expertise in these two different classes. I'm going to start off by presenting empirical evidence that support the argument for separability of face and word recognition systems. So this assertion of independence, as you have already heard, is strongly played out in the neuropsychological literature, with a lesion to the right occipitotemporal cortex resulting in prosopagnosia, and a lesion to the left occipitotemporal cortex resulting in a word recognition deficit, pure alexia, in premorbidly literate adults. There are also electrophysiological data that support the separability, with a large negative waveform deflection over the right hemisphere for faces and over the left hemisphere for words. And as I have already shown you, many functional imaging studies that show face-specific activation in the right hemisphere and word activation in the left hemisphere. However, close inspection of these imaging findings suggests a different organization. So even in the paradigmatic study of the fusiform face area, where a rectangle is superimposed on the right hemisphere activation, there is actually also activation in the left hemisphere, albeit not as strongly. And this is true in all of these studies where we see bilateral activation, but stronger in the right hemisphere than the left hemisphere for faces. This is also true in the case of words, where we see binary activation as well. So in the paradigmatic study that mapped out the visual word form area, in this case a PET study, there is actually also activation in the right hemisphere. And we see in these other studies as well bilateral activation, just more strongly in the left hemisphere for words. So while this bilateral activation is certainly compelling and provocative, one of the things we still don't know from aggregating these studies is what the relationship is between the mechanisms that support face recognition and those that support word recognition, because these studies only ever show the viewer one of the stimulus classes. So a number of years ago, in a study with Uri Hassol and Rafi Malat, we had participants in the magnet viewing both words and faces. You're looking down here in the center on the inferior surface of the brain, much like the images that you've seen previously, just to make it clear. If I took a wire cutter and sort of somehow cut off my head and turned it over, you would be looking right down onto the base of the brain. This is, so we're looking down onto the ventral view. On each side is a flattened map of cortex. And this is particularly useful because there are bits of activation that are curled under this hemisphere that on the flattened map can be fully observed. So in this study, we were able to replicate the bilateral activation for words, greater in the left hemisphere as shown here in the mauve color than in the right hemisphere, and for faces with greater activation in the right hemisphere shown here in orange compared to the left hemisphere. So this is particularly interesting because we are seeing abutting areas in the two hemispheres representing words and faces, but with this weighted or graded asymmetry. It's also interesting that these regions seem to be really homologous, because if we take the peak of the centroid of activation and show it in Taylor-Rack coordinates, these regions are actually very similar to each other, kind of compelling us to think about the interdependence between face and word recognition in equivalent kinds of cortical territory in the two hemispheres. So we have bilateral weighted asymmetry, and this should be a compelling account of the neural substrate for face and word recognition. But of course, we also want to understand, where does this organization come from? So together with Eva Dundas, a graduate student, David Plant and I hypothesized that the weighted asymmetries emerge over competitive and cooperative interactions within and between hemispheres that play out over the course of development. So in young children who have not yet learned to read, both hemispheres are being increasingly tuned for face recognition. However, once children begin to go to school and through early adolescence, as they become exposed to orthography, so the prediction is that you would see representations in both hemispheres for orthography, but to a greater degree in the left hemisphere because of the pressure to have close connections between extrastriate visual areas and language areas, which obviously are in the left hemisphere in the majority of the population. And last, in adulthood, we see a refinement of this bilateral activation, with word recognition becoming firmly embedded in the left hemisphere, albeit not exclusively so, and face recognition being largely shunted out into the right hemisphere in the adult profile. We assessed this hypothesis with 3 groups of participants, 24 participants in each of these groups, in really this almost the simplest experiment you can ever think of. They sat in front of a computer screen, a fixation appeared, and then a single face was shown for a relatively long duration, long enough so even the youngest children are able to encode the stimulus. Then a brief fixation so that they don't move their eyes, and then a second face appears, either in the right or the left visual field. Participants are making a key press, whether the second face is the same or different from the first face, and there are blocks of faces, and there are blocks of words. Here are the findings from the adults. Accuracy is plotted here on the y-axis, and as you can see, there is a superiority in recognition over in the left visual field, i.e. the right hemisphere for faces, and in the right visual field, i.e. the left hemisphere for words. Notice also that face and word recognition in the non-preferred hemisphere is rather good, consistent with this bilateral weighted account. When we look at the results from the adolescents, their overall accuracy is very similar to that of the adults. But interestingly, we only see a superiority for words in the right over the left visual field, and no lateralization differences for faces. And then in the young children, although their overall accuracy is much lower than in either of the other 2 groups, they too, like the adolescents, are only showing lateralization for words and no hemispheric superiority for faces. This suggested to us that there is an earlier emergence of word lateralization, and then perhaps the pressure to read then drives the emergence of face lateralization through these kinds of competitive and cooperative processes. So in these same individuals, just the young adolescents and the children, we have plotted here their reading proficiency as measured on an independent measure. This is a standardized reading comprehension test. And on the y-axis, we have the degree of lateralization for faces, so the extent to which there is superiority in the left visual field, right hemisphere, over the right visual field, left hemisphere for faces. And as you can see, there is a nice correlation between them. So the better the reader you are, the more you have lateralization for faces, suggesting an intriguing relationship between those. We also collected ERP data, but we did it only in the adults and in the young children. I'm showing you here the peak of the amplitude of the N170 activation, and I've plotted negative going upwards here to be consistent with the orientation of the behavioral data that I've just shown you. So the higher the negative value here, the better the activation. So you can see in adults greater activation over right hemisphere electrodes for faces than left hemisphere electrodes, and the converse is true for words, with greater activation over the left electrodes for words compared to faces. And intriguingly, in the young children, we only see, as we did in the behavioral data, the advantage, the hemispheric superiority for words over the left than the right hemisphere, and no significant difference between the faces. And so these findings are consistent with an account that it is that over the course of development, this organization is a manifestation of the underlying cooperative and competitive mechanisms that eventually shake out into the adult hemispheric organization. As a means of exploring further these cooperative and competitive processes, Nick Blausch, a graduate student who worked together with David and me and a postdoc in my lab, collected data from 55 adult individuals on both face and word recognition, but also recognition of many other stimulus classes that I won't describe today. Activation for faces is shown in red. Again, I'm showing you flat maps. And activation for words are in blue. And unsurprising at this point, we see both blue and red in both the right and the left hemisphere. But notice, of course, that in the right hemisphere there is more red than blue, and in the left hemisphere there is a preponderance of blue over red. Also, interestingly, in the left hemisphere, we're able to elicit activation to written words in Broca's area, and this co-localization of the visual word form area with Broca's area is thought to be one of the results of this cooperation within hemisphere. So you may be wondering, why would we ever see a neuropsychological deficit like prosopagnosia in these cases and pure alexia in these cases? If we've got representations in both hemispheres after a lesion, why doesn't the other hemisphere simply step in and take over the function? This drove us to look very carefully at the individual profiles in the sample. So most of the 55 individuals show this bilateral pattern of activation that is reflected in the average group map. But there are individuals who show a more unilateral pattern, so faces only in the right hemisphere and words almost exclusively in the left hemisphere. And it is my conjecture that these unilateral cases are going to be the future pure alexia and prosopagnosic individuals. And in fact, it may be unilateral cases like this that are found in Randy Starfeld's studies who show either association or dissociation between faces and words. In fact, this unilateral pattern is rather rare in our sample, in 2 to 3 of the 55 individuals. And of course, these neuropsychological deficits are also rare in the population at large. We went on to examine the relationship between the selectivity for words in the left hemisphere and faces in the right hemisphere over the 55 individuals. And again, you can see a very nice relationship between them, with a greater the activation for words in the left hemisphere correlating with a greater activation for face selectivity in the right hemisphere. So you've heard the argument for the competition between words and faces now repeatedly. These results are consistent with the data from Behrmann et al., as already introduced, I think, in Alex's talk, in which they reported face selectivity in the left hemisphere in the illiterate individuals. And then, interestingly, as these illiterate individuals began to acquire literacy, so you begin to see a reduction in this face selectivity in the left hemisphere and the emergence of the visual word form area, but also an increase in the right hemisphere for face recognition in these ex-illiterate individuals. But not everything in the world aligns perfectly. And there are some challenges to this particular view. For example, Lort et al. Have argued and have shown data, don't just argue, you actually show the data, that the left hemisphere word selectivity is a recycling not of face selective cortex, as we've argued, but of limb selective cortex. And then there are a number of other studies that have shown that word selectivity appears to emerge in regions of cortex that were not previously tuned to any stimulus type. So clearly, there remains much to be done to compare and contrast these different accounts. To what extent is this now clearly articulated hemispheric organization rigid? And what are the consequences of damage to the system and the possibility for plasticity or malleability of this hemispheric organization? So for the next short while, I'm going to tell you about the malleability, the reorganization or plasticity in patients. So we already know that after even a small circumscribed focal lesion to the right or the left hemisphere, prosopagnosia and puralexia respectively can occur. I was particularly interested in understanding what are the consequences of lesions, but in children who have the greater opportunity for plasticity. So I chose to study children with under extreme circumstances, and these are children who, in contrast to the normal brain, only have a single hemisphere, in this particular case, a resection of the left hemisphere and a preserved right hemisphere, and here we have the converse situation. These dramatic surgeries are done for the management of drug-resistant epilepsy and are actually highly effective in eliminating seizures in 80 to 85% of the cases. People always ask me, so what happens with this cavity now? It's filled with cerebrospinal fluid and is essentially, dormant from a functional point of view. So, unsurprisingly, these children have homonymous hemianopia. I'm not sure you can see this so well, but on visual field testing, you can see that those who have left hemisphere resection have a right visual field hemianopia, and those with a left hemisphere resection have a right visual field hemianopia. It's hard to keep the rights and lefts separate here. Notwithstanding the dramatic resection and the homonymous hemianopia, the consensus in the field is that the majority of post-surgical children have a good visual outcome, which is exactly what I'm interested in. And so together with Michael Granovet, who is a graduate student in my lab, we put together a slightly different version, but of the same sequential discrimination task that I showed earlier. So there's a fixation, the face appears centrally, another brief fixation, and the second stimulus appears centrally as well. We wanted to capitalize on foveal vision most of all, but we were then concerned that it may even be the case that the hemianopic children we're testing may have some compromise of central vision. And so we had all the participants perform 2 versions of the experiment, this one at fixation and a second one in which the probe stimulus appears only in the intact visual field and either in the left or the right visual field of the matched controls. We were able to recruit 40 childhood hemispherectomy patients, and we did this over the course of the pandemic because we miraculously refined a remote testing procedure where the patients would log in to our server and then be able to perform the experiment. And here are some examples of the scans of the patients. They've asked me to keep the lights high because they're recording, but I'm just going to turn them down for a moment so that at least you can get a sense of what these lesions look like. I think it takes a few minutes for the lights to go down. So again, these are the axial slices, much as the ones that I've shown you previously, and we have 40 such individuals. And here are the data. So this is for faces and this is for words. On the y-axis is accuracy. In blue are the results from the controls. Every dot here is a different participant. And we have light dots because we had young controls and dark dots for older controls because we had both younger and older patients as well. So the controls are performing about 95% accurately for both faces and words. This is obviously a trivial task. Here are the data from patients with just a left hemisphere or patients with just a right hemisphere. The mean accuracy is about 85% correct, statistically inferior to that of the controls, but remarkably similar, irrespective of whether you only had a left or only had a right hemisphere. And this is true not only for faces, but also for words. So with just a single hemisphere, so you remove anatomically 50% of the brain, and it's still a single hemisphere yields 85% accuracy. It's a pretty good bargain. Especially if you're relieved of seizure activity. And also, importantly, in just the single developing hemisphere, it doesn't make a difference whether it's the right or the left hemisphere. This kind of accuracy level can be accomplished. What does the neural organization of that single hemisphere look like? So, again, together with Michael Granoveta, we recruited a new set of 9 occipitotemporal cortex resections, much like the patients that I've just shown you. We also had 21 matched controls, and we recruited a second patient group who also had resections, but outside of occipitotemporal cortex. So any results that we see are not because of a resection per se, but because of the occipitotemporal resection. And we put them through their paces with this topography selectivity experiment. And here are the data. So this is the amplitude of category selectivity for faces, objects, and words. And this is the left hemisphere of the typically developing controls and their right hemisphere. And you can see that there is this greater amplitude in the right than the left hemisphere for faces, and the left over the right hemisphere in the controls, and bilateral equivalence for objects as is sort of the standard finding. Here are the data from those with resections outside of occipitotemporal cortex. They fall within the normal range. But dramatically, the patients with occipitotemporal resections, or hemispherectomies in most of these cases, also are showing, just in their single hemisphere, category selected amplitudes that fall within the distribution of the control, of both control groups. So this is really interesting. A single hemisphere appears to be able to accommodate both of these functions. But of course, when we operate in the real world, recognition is not done at the category level. We don't go around saying that's a face, that's a word, that's an object. We really recognize at the individual level. So we need to know which of these two fairly similar looking faces is my brother, for example, and which of these two fairly similar looking words is the word knight. And so I will now assess the nature of the individual level representations in a single hemisphere, with the expectation that we might see some breakdown under these conditions. So we're able to exploit a manipulation that goes as follows. This is the logic. So a participant lies in the magnet. We show them a whole stream of faces, of different faces, one after the other, and we measure some amount of bold activation. We also can show a run of stimuli that is just the same face, repeated over and over again. And under those circumstances, we see a reduction in the activation profile, presumably because the neurons have now become habituated or fatigued by the repeated presentation of the same face. This is the adaptation effect, so different minus same, the difference between the different run versus the same run is going to reflect this individual level representation. And we perform the experiment on novel faces, novel words, so non-words, and also novel objects, greebles, that you also heard about. So plotted now on the y-axis is the magnitude of repetition suppression, really the difference between the different face runs compared to the same face runs. And we can see in all of these categories, the typically developing controls show repetition suppression to the same stimulus. This is also true for the control patients. And staggeringly, this is also true for the individuals with a single occipitotemporal cortex. So we were both rejoicing, but then also concerned, because we show even normal repetition suppression magnitude, sort of suggesting that one hemisphere might suffice for recognition, but I've already shown you that recognition is statistically inferior to that of the controls. So how do we resolve this kind of conundrum? Well, one possibility is that each hemisphere contributes slightly different information to the recognition. A single hemisphere can get you 85% of the distance, but two hemispheres are really needed to obtain fully normal expert recognition of these two stimulus classes. In the next few slides, I want to show you how these neural changes occur within a single hemisphere. Mostly I've shown you cross-sectional data, and I'm now going to show you some longitudinal data that allow us to map the trajectory of this plasticity over time within an individual. So here is one of these individuals. I'm not violating confidentiality. After we published a study, the press interviewed him and his family. His name is Tanner Collins. He had a tumor, a DNET tumor, in the left occipitotemporal region that was identified through having seizures at around age 4. And he went on to have a gross resection of the occipital and temporal cortices in the right hemisphere. While we might have expected him to be prosopagnosic, given the resection of the key face regions in the right hemisphere, He assured us that he can recognize faces perfectly, and in fact that was true empirically. And he also convinced us that he wants to become a neurosurgeon himself. So actually 2 weeks ago, he came for yet another follow-up session. I have not analyzed the data from that. But he seems to have changed his mind and has decided now that neurosurgery is not for him. He wants to become a mathematician instead. So here are the longitudinal data. This again is the resection. This is his code name that we use in the literature to cover his true name. And this is the timeline of the longitudinal study. He had his first seizures, he had his lobectomy at 6 years, 9 months, and then we scanned him and did behavioral testing over sort of roughly 6-month intervals up to age 13, and as I say, we're continuing to scan him. What I'm showing you here is the result of the imaging of a region in his single preserved occipitotemporal left hemisphere region. This is an inflated view of his brain, of the surface of his brain. And you can probably appreciate this big chunk that's been taken out here in the right hemisphere. Each of these plots is one of the scanning sessions, and this is the 3D volume of this particular area. Every little dot here is a different voxel in the brain, and they are colored according to how face selective they are, shown in red, or word selective they are in blue. And so you can probably see relatively early on, there isn't a great commitment to either faces or words. It's sort of in the mid-range of these T values. But you can begin to see then the emergence not only of face selectivity in red, but also the emergence of word selectivity in a cluster abutting the face selective regions. And just to show you in greater detail, you can appreciate the strong face selectivity and word selectivity in these neighboring regions in the single occipitotemporal cortex quite dramatically. We had another individual case whom we scanned longitudinally. This is patient TC. She's somewhat more interesting in the context of this hypothesis because she had a left hemisphere resection. So the visual word form area is essentially missing in this individual. And because we know about the co-localization of the visual word form area with language, it becomes somewhat uncertain what the outcome is going to be here. She does have left hemisphere language dominance in imaging. And again, we've got a longitudinal timeline here with many of these category localizer experiments over time. Now let me show you the data. So we only had 3 sessions from her. You can see the resection here. And again, this is the volume, the 3D volume of this region with each voxel showing the extent to which it's face or word selective. She only had her surgery somewhat later in life, and so language was firmly embedded in the left hemisphere at age 13. And you can see that at this point she had fairly strong word selectivity. She was a non-native English speaker, and so she had begun to acquire English a little later in life. And you can see, perhaps related to that, that we only see the emergence of word selectivity rather later than we might expect to see in the typical child. And again, we see the abutting clusters of face selectivity and word selectivity in the single preserved, in this particular case, right hemisphere, even the visual word form area emerging in the right hemisphere. So you might be wondering whether plasticity is ubiquitous. It's like everything we touch seems to show plasticity. But I will show you that that's not actually true. With Vlad Eisenberg, who was a postdoc in my lab, we again recruited a new group of participants with either right resection, preserved left hemisphere, or left resection, preserved right hemisphere. So the first thing that we did is look at whether or not we could replicate this plasticity in ventral visual cortex that I've already described. So we had participants, this is sort of the cartoon of greater face selectivity in the right hemisphere and greater word selectivity in the left hemisphere. It doesn't suddenly become backwards. Words are not, I hope you can see here, this is backwards. I just did the random flip from left to right and didn't take into account the fact that words have a particular order, so I'm sorry for that. So this is the neural selectivity for words and for faces. This is for the left hemisphere of the controls in red and the right hemisphere of the controls in blue. And you can see this nice hemispheric difference for words and the converse difference for faces. And each square here is one of the hemispherectomized individuals. And all of their values, and this is the mean of the group, fall within the normal distribution for both words and for faces, irrespective of whether they have a preserved right or left hemisphere. So it was a happy day to be able to replicate our own previous findings. But we wanted to go beyond this, and we wanted to look at dorsal cortex, which is also a visual area. And there are interesting data in typical individuals that show that there is greater activation in the left hemisphere simply from observing tools than in the right hemisphere, perhaps because of the right-handedness that goes with the left hemisphere motor function. And so you can see here in the control individuals a bias to greater left hemisphere activity for tools than in the right hemisphere. And the converse finding is in global form perception, which I've sort of mapped out here just by showing you the spatial relations between the different parts of an object. And for global form, one typically sees greater activation in the right than in the left hemisphere, or this is a bit less dramatic than for tools. And so we looked at this in the patients as well. And here are, again, the individual patients with the right and the left hemisphere resections. And there is these individuals are showing very little plasticity, sort of almost no differential response in the right and the left hemisphere, and really low activation for both of these dorsal functions. So the presumption here is that dorsal cortex might have an earlier sensitive period, and so by the time these kids come to resection, dorsal cortex sensitive period has already closed, and there is less opportunity for plasticity. In contrast, ventral cortex has a prolonged developmental trajectory, and so there is still opportunity for reorganization following surgery. Okay, so plasticity is not ubiquitous. So in the last short part of my talk, I'm going to change tack a little. So instead of focusing just on words and faces and just on ventral or dorsal cortex, I'm going to show you data that evaluates the large-scale reorganization in the preserved hemisphere. And the key question here is, what is going on in the preserved hemisphere that permits the accommodation of these other functions? So the way in which we did it, this is Sophie Robert, who led this study as a graduate student in my lab. We opted for the most naturalistic experiment that we could think of, which in imaging is lying in a magnet and watching a movie, extremely naturalistic. And we had all of the children watch this movie, Escape to Witch Mountain. It's an old movie. We chose it specifically because we didn't want anybody to have more frequent exposure or familiarity with the movie. So participants watched a part of the movie before they got into the magnet, and then we continued the movie and did 2 runs of an 11-minute movie clip that followed on from what they had been viewing previously. And so the question is, how are the functions from the resected cortex now accommodated in the preserved cortex? So we parcellated the preserved cortex into 8 parcels or 22 parcels or 180 parcels using either the Glasser or the Schaefer atlases. And this is a bit of a Goldilocks problem. The 8 parcels is a little too coarse to see real reorganization in that hemisphere. The 180 parcels is somewhat too large. I'd have to go through 16,110 pairwise correlations to show you that. We have actually published the 180 and the 8 data along with the 22 if you're interested. So I'm just going to show you the sweet spot here, 22 parcels. And the way that we do this experiment is for every voxel in the intact hemisphere in the patients and in the matched hemisphere of the controls, we extract the time course in each voxel over the duration of the movie, and then we can look at the correlation of voxels within a parcel compared to the pairwise correlations between parcels in this connectivity matrix. And we do this for 7 patients with just a left or just a right hemisphere, and also the corresponding hemisphere in the matched controls. So the analysis, again, just to be clear, is we're going to look at the pairwise voxels within each of these 22 parcels, and we're also going to look at all pairwise functional correlations, really, between all of these parcels. So this is really a correlation analysis and is often thought of as functional connectivity. We elected to use a multivariate approach to this, giving us more precision and power in this kind of analysis. And I'm going to show you the data on these chord plots. So this is I'm going to show you alterations in the connections in the left hemisphere controls compared to the preserved left hemisphere of the patients, and again, just the right hemisphere of the controls against the preserved right hemisphere. So the more green a region on the circumference is here, the greater the reduction in the patients relative to the controls. It's kind of a bit of a double negative. So, sorry, let me just say that again. The more green something is, the poorer the connections in the patients. The more purple something is, the greater the connectivity in the patients compared to the controls. So the circumference only plots the within parcel changes, and for convenience of plotting, I've taken these 22 regions and kind of just aggregated them into somewhat intuitive groupings: frontal regions, vision area, motor area, etc. So in both hemispheres, both the left and the right comparisons, we see a reduction, largely shown here in green, in the left hemisphere, and to an even greater degree in the right hemisphere. It's also kind of interesting that there is some preservation. This is largely going to be associated with some preservation of visual cortex, and in a moment I will show you now the alterations between parcels. So these are all the alterations that are tending towards significance in the contrast between the patients and the controls. Again, green means that there is greater reduction. When one corrects for this multiple comparisons, you eventually only get a subset of significant results, with a reduction in between parcels of 9% in the left hemisphere and 22% in the right hemisphere. So we do see changes, significant changes, in both the within and between parcels, in both the right and the left hemisphere, but to a greater degree in the right hemisphere. And I have no doubt that you're already all thinking about why this might be. And presumably, what happens here is that there is strong pressure to accommodate language function in the right hemisphere. And language is sort of the most fundamental cognitive ability, sort of the paramount ability. And so the pressure to accommodate language gives rise to major changes in the right hemisphere. And that same pressure is not true of the left hemisphere that accommodates the right hemisphere functions, which does not include language. Okay, in just my last few minutes, I'd like to share with you a new avenue of work that we've been doing. So I've shown you so many functional imaging slides and also some behavioral data. Functional MRI is sort of great. It's like a gift from the cognitive neuroscience gods. However, it is a truly blunt instrument. Each voxel contains more than a couple of 100,000 neurons, and the temporal resolution is obviously very poor because it's contingent on the very slow and sluggish hemodynamic response. So we really would like to try and find something else to begin to get greater purchase on these questions of hemispheric organization. So in the last maybe year, 18 months or so, to address questions of representations and temporal dynamics with greater resolution, Xu Yuling, a postdoc in my lab, has led an effort to examine the neural signals that come from the children who undergo stereoencephalography. So let me explain this to you. So prior to resection, children with drug-resistant epilepsy are brought into the hospital, and the neurosurgeons implant these deep intracranial electrodes into the probable epileptogenic zone, and they monitor the seizures in this area so as to be able to demarcate the boundaries of where the resection is going to take place. These are very thin wires that are projected directly into the brain, and on each wire, which are called shanks or shafts in this field, you can see that there is a whole slew of contacts or electrodes. This is particularly interesting, this F over here is particularly interesting because this shaft goes right into temporal cortex in this individual. Here in the same individual is an illustration of this. So this is the post-implantation CT scan, and you can see all of the electrode contacts in this individual. So they're usually something like 14 to 18 shafts that are inserted, and each of these have 16 to 20 contacts. So on average, we collect data from about 200 channels simultaneously. So I hope you can see the electrode locations here. We now have data from 1246 electrodes in 6 patients. We actually now have 9 patients. We just haven't analyzed the data. And I have here mapped all the electrodes to the template MNI brain so that you can see its localization. So for me, fortunately, temporal lobe epilepsy is the most common form of epilepsy in young children. And so we see a huge slew of electrodes in temporal cortex, which is good for my particular area of interest. So I'm only going to show you our preliminary data, which assays category selectivity on these intracranial electrodes. But we have a simultaneous project in which we are comparing the ventral and dorsal neural dynamics because there are many dorsal electrodes as well. So this is the same kind of experiment. You just show visual stimuli. The timing in this kind of experiment is as follows. Firstly, there's a pre-stimulus onset, so we're recording from all the channels during the baseline. And then each stimulus in this run of common objects has an object appear for 800 milliseconds, blank for 200, object in this oscillatory pattern. And what I'm showing you here on the y-axis is each of these contacts on this F electrode. To the left of this vertical bar is the baseline period, and then the stimuli start appearing. So for faces, places, objects, scrambled images, and words. And what you can probably see with the naked eye is that there is an increase in broadband power once the stimuli start appearing. And moreover, the temporal dynamics of the stimuli follow the duration of the exposure. So we get strong responses for the 800 milliseconds when the object is on, and then a break in the fixation condition, and then again this sort of very driven, coupled response between the broadband power and the nature of the stimulus presentation. So the first question that we set out to answer is what is the nature of category selectivity on these electrodes? So the way that we have addressed this first question is as follows. Each of these is a single individual. So we train a machine learning classifier on 80% of each individual's data, and then tested its ability to predict the category in the remaining held out 20% of the data, and then we basically permuted through all of this, and we've got very, very many data points for each of the individual children. And as you can see here, whereas chance is 20% because we have 5 categories, all of those who have a little red dot above them is where the classifier accuracy is significantly above chance. So this was our first foray into this field, and we were particularly enthusiastic about the fact that we could actually predict the category of the held out data. We are now in the process of doing much more fine-grained and much more detailed analysis of the temporal dynamics, as well as the dynamics as a function of age. These kids are all slightly different ages. It's not a very common procedure. They are quite rare cases, but we're roughly seeing one every 2 to 3 weeks, and so I'm hoping that we will have enough data in the not too distant future to be able to have a sort of much better foundation for the empirical work. Okay, so it's been a long journey, but let me review for you what we have covered. So instead of thinking about the hemispheres as either the face or the word hemisphere, it really behooves us to understand that there is a bilateral weighted asymmetry across the 2 hemispheres. I then suggested that this mature pattern comes about through the cooperation and competition that ensues over the course of development, but that there are also individual differences with some unilateral patterns or more unilateral patterns, potentially because of differences in the cooperation and competition. And we had also collected diffusion tensor imaging data from all of these participants so that we can now begin to explore whether or not these individual differences might come about because of, for example, the volume of the corpus callosum. So the larger the corpus callosum, the more the 2 hemispheres would be able to talk to each other. Whereas those with individual differences might just have very small corpus callosum and sort of pressuring a single hemisphere to take on a single function. We then looked at the extent of plasticity for these organizational profiles, showing that in fact in the hemispherectomy cases we can see face and word recognition in a single hemisphere. Using neuroimaging, we can show representations at both the category and the individual level. And in the longitudinal data, we can see how over time these category selector voxels for faces and words come to abut each other and cluster in a single hemisphere. We also showed that plasticity is not ubiquitous, and I think that this is an important result, even though it's a bit of a sort of null result. We don't see plasticity in dorsal cortex. It serves as a really strong contrast with the ventral plasticity that we've been reporting. And we also looked at the preserved hemisphere, at the within and between parsal changes, where there are many more within than between parsal changes, and many more in the right hemisphere than in the preserved, many more in the preserved left hemisphere than the right hemisphere of the patients. So I'm going to end off here by pointing out these particular graduate students and postdocs whose work I mentioned in my talk today. I'd also like to recognize my collaborator, friend, spouse, David Plant, my very good colleague, Christina Patterson, the Director of Epilepsy at Children's Hospital in Pittsburgh, and also Monica Jones, who is the founder and director of an agency that she started called the Pediatric Epilepsy Surgical Alliance, that she started after her son Henry was born with hemimegalencephaly, so one teeny hemisphere that is very malformed and one intact hemisphere. And this organization has partnered with us and allowed us, I think permitted us to recruit such a large number of participants, but they've also served so many families and advocated for these, the children with hemispherectomy, and that's been an astonishing contribution. So I will stop here and thank you again for this wonderful opportunity to present a Bartlett lecture. I'm truly humbled. And I am so grateful for the wonderful talks in the symposium. You all made it sound like my work is so much more exciting than the way that I've sometimes thought about it. So thank you all so much. I'm happy to take questions, but we mustn't miss the bus.
No. Any questions?
Yeah, thank you very much for this excellent talk. I learned a lot, but also because I'm not in the field. My impression is you clearly showed the fact that visual language processing is lateralized to the left, but I missed explanation for why it is. So during your talk, you briefly alluded to the within hemisphere support as one possible explanation for why visual word processing is in the left hemisphere, not in the right. And I take that to mean it's because speech processing, Broca is left already, the children can speak. And another thing you said near the end is that there is some pressure, which I didn't quite understand what that means. So can you explain the mechanism that drives this lateralization for visual words into the left? For example, do you think that spatial frequency tuning of the hemispheres and the need to have this fine analysis of the words that have very similar envelopes is a key factor? That's my question.
Great, thanks for the question. There's sort of two parts to my answer. The first part is, why is language in the left hemisphere? Because it could be a coin flip for each individual. So why is language in the left hemisphere? Well, I don't think anybody really knows the answer to this, but let me share with you one of the existing theories, and it'll give you a sense of how sort of primitive some of our understandings are. So the particular theory is called the Peloponnesian War theory, and the claim here is that during the Peloponnesian War, those individuals who were right-handed had their spear in their right hand and their shield in their left hand covering their hearts. And they survived the war compared to those who had hearts exposed. And it is that subgroup of individuals with right hemisphere dominance and presumably left hemisphere language dominance that now that is the paradigmatic profile, and then genetically propagated their species in the majority of the population are right-handed today. So I don't think we know. There are lots of theories about why language is left hemisphere dominant. Some people talk about the ability of neurons in the left hemisphere to be able to monitor very rapid changes as is needed for auditory processing. So let's just assume that language is in the left hemisphere. And now, what is it about language that is going to influence the hemispheric organization in extrastriate cortex for visual stimuli? So the claim here is when we read, we not only are trying to analyze the visual features of the input, we're trying to understand what we are reading. And so we do want to have language areas as closely coupled to extrastriate visual areas as is possible. And it is this coupling, the sort of the pressure to keep connection lengths relatively short, mostly for metabolic purposes, it's this coupling that is essentially driving the preponderance of visual word activation into the left hemisphere, and then kind of by competition, faces get shunted out primarily, albeit not exclusively, into the right hemisphere. Now, we've also looked at things like the spatial frequency tuning of, not just us, many people have looked at spatial frequency in the two hemispheres and have argued that the left hemisphere has got a higher spatial frequency tuning than the right hemisphere, and other people have not been able to show that. We also have looked at the spatial frequency tuning of the single hemisphere in the hemispherectomy cases. So is that single hemisphere more like the tuning of the right hemisphere or the tuning of the left hemisphere, or the sweet spot between them? We're not quite finished with the experiment, and generally I'm a bit tentative in making statements about things that I don't definitively know yet. But at least there is a sense that there is sort of tuning for mid-frequency in the single hemisphere of the patients. And then, of course, the question is, well, where does that come from? Thank you.
Any other question?
There's one here.
Sorry.
So, do the relatively rare people with hemisphere represented in the right hemisphere, language represented in the right hemisphere, are they descended from Peloponnesian warriors who had a shield on the other side?
Yeah. So it's not solely environment that has given rise to the handedness and hemispheric organization in the population. In fact, the Peloponnesian War theory obviously is somewhat laughable in the state that I explained it. There's a genetic factor as well, and there are lots of people who argue for different genetic sort of flips of the coin that result in language in either the left or the right hemisphere. And it's actually hereditary. So if you look at monozygotic versus dizygotic twins who have right hemisphere language, you see it greater in the mono than in the dizygotic twins. So there's definitely a genetic aspect of this as well. I'd love to see some of these resection patients with right hemisphere language and to understand the consequences of that. Yeah.
It's such a small sample.
It's very small, but I have my eye out for it all the time.
Great work. Thank you very much for answering the questions and for such a brilliant talk.
Thank you so much.
Thank you again on behalf of all of us.
Thank you.